Yeah, thank you all for being here. Okay, let's start. Nice kind of gong sound. Yeah, thank you all for being here. It's a pleasure to kick off the first Macroevolution session of the conference. My name is Keiffer Williams. I'll officially start now. I am a PhD candidate at Clemson University, and on behalf of my co-authors, including three hardworking and talented undergraduates, Lanier, Josh, and Fong, they helped me segment a lot of micro-CT scans for this presentation, including this parrotfish. I'm going to be sharing with you some of my dissertation research, which is addressing the degree to which herbivory and other dietary guilds are associated with tooth complexity in reef fishes. And starting out, I want us to just think very broadly about teeth. So I think teeth are essential tools for understanding vertebrate evolution. They're important for form-function relationships, as we can see here with these teeth of the seal used for filtering krill from the water, or the teeth of this alligator used for crushing up the shell of a crab. They're also important for inferring diets and living in extinct species. We can see here in this elephant, we can see what an elephant's eating and compare that with some of the dentition of an extinct mammoth and sort of infer what that mammoth might have been eating in its prehistoric diet. And so although we've recognized these qualities about teeth for some time, generally studies of teeth have been restricted to qualitative descriptive studies. That's changed in the 21st century with rapid advances in micro-CT scanning technology. A method known as tooth-surface complexity has been established, and it allows for non-homologous dental structures to be compared and tested for correlations with ecological correlates such as diets in mammals. And so across studies of carnivorans, rodents, and chiropterans, as well as several other mammal groups, a general pattern has emerged that herbivores generally have more complex diets than other dietary groups. And this pattern is thought to be attributed to the tough material properties of things like leaves, roots, and shoots, which require a bit more processing prior to digestion in the system. Furthermore, this has also been extended to reptiles, tooth-bearing reptiles, even more recently, turtles, which do not have teeth. And so even across non-toothed turtles, the same general pattern has been established, herbivores being more complex. I will point out duriphages have also been found to show similar complexity to herbivory as well. And overall, this suggests herbivory imposes similar evolutionary constraints on tooth complexity across two distinct clades. However, as I've alluded to in my intro slide, I'm interested in fishes. And for some reason, we still lack a really, even a basic understanding of whether fish teeth track ecological correlates over macroevolutionary timescales. It's interesting to consider that, given that fishes make up nearly half of all vertebrate diversity. So there's a lot of understanding that we're lacking there. Part of this lack of understanding is because, fishes are a little fishy with their dentition, a little challenging. They exhibit a high variation in rows and number of teeth across the jaw, and this is true for even very closely related species. And maybe even more interesting, nearly all bony fishes exhibit a two-jaw system where the oral jaws are responsible for prey capture, frontal jaws are responsible for prey processing. And so if we compare this to the dentition of a single jaw system, such as a mammal here, selection is acting on just a single jaw with both prey capture and processing occurring in the single system. Whereas fishes, they're partitioning into two different processes of capture, prey processing. And so selection is likely acting in two kind of different ways across this two-jaw system, which might be influencing patterns of tooth complexity. And so some great groups of fishes to kind of explore a bit with tooth complexity are the wrasses and damselfishes. These are some iconic, hyperdiverse, ecologically diverse groups of fishes that include the iconic parrotfish with its fused beak dentition, the tube snouts of the bird wrasse. We have clownfishes. We have really anything and everything in between. Quite a morphologically, ecologically diverse group of fishes to explore tooth complexity in. One thing I want to mention is that work over the last few decades in fishes has shown that categories such as herbivory are a bit more of a catch-all term that can actually encompass numerous ecological processes. And so to recognize this functional complexity, we're going to be using different tooth functional categories that we established to try to capture some of those ecological processes that are occurring on coral reef ecosystems. These include things like sediment excavators and sediment scrapers. These are almost exclusively parrotfishes, as you can see there with their fused beak dentition. Excavators are biting off chunks of coral sediment and actually breaking it off the substrate and processing it. Scrapers are contacting the coral but not actually breaking it off. Everything ranging down from algal croppers and then even down to shell crushers are some of our groups. As you can see, if we put these into the more kind of classical bins of herbivore, omnivore, planktivore, generalist carnivore, you can see how these ecological or these functional categories are better getting at those ecological processes going on. And so with that, we're asking a fairly straightforward question here, and that is, how does tooth function influence tooth complexity in reef fishes? And again, we're interested in this in part because of that two-jaw system. Tooth complexity might be partitioned differently across the jaw in these two-jaw systems versus the more standard one-jaw system in mammals. So to do this, we did a lot of micro-CT scanning. We scanned 115 species at Rice University in Houston. We segmented out the oral jaw, so we're just focusing on the oral jaw for this study. And then looked at either the left or right side dentary, really whichever one was more complete or easier to segment. And then we looked at all the teeth and tooth surfaces that were present on that jaw. So this method I've been talking about, tooth complexity, more specifically it's called orientation patch count rotated, or OPCR. The way this method works, if you take a conical tooth, viewing it from a lateral orientation here, the method starts by viewing a tooth from a top-down or occlusal view. So here, that little black dot, imagine you're the prey item, looking down the tip of this tooth. And once it's in that position, it then breaks the tooth up into different patches. And here with a perfectly conical tooth, we have 8 equally sized patches. And a patch is just a group of 2 or more pixels in the scan that are oriented within the same orientation. So if you take something that's more complex, such as a rodent molar, what they found in the original study that pioneered this method, rodent molar is going to be a lot more complex than the basic conical tooth. So they found there were 122 patches total on this rodent molar. So what that tells you is that when you have more patches, you have a higher tooth row complexity. So PCR tells you patches, and having more patches means you have a more complex tooth or tooth surface. Another thing I'd like to point out with this method, you have to focus on resolution as well, and so we account for resolution using mesh triangles per tooth. And we do this to try to account for variation in tooth size across the jaw. So let's break it down a bit here. When you run OPCR in the jaws, here we have 151 patches encompassing all the teeth in the jaw. We ran this at 1,500 triangles per tooth. So what that means is that we approximately assign 1,500 triangles for each of the 18 teeth along this jaw. And so you can do this at different resolutions. We did it at 1,000. We also did it at 2,000. And what you generally see is that complexity will increase at higher resolutions. And so this has also been shown in studies of mammals use this method, reptiles as well. And so we're also seeing the same pattern in fishes. And you can generally see that here. If you go from 1,000 to 2,000, your patch count is increasing a bit each time. So I just want to point that out for you all. I'm going to be presenting your results just on the 1,500 triangles per tooth, but just bear in mind that these results are generally consistent across the different groups. So once you have your resolution set, you can then run the OPCR analysis. That's what we see here with 151 patches. So that's the OPCR total. That's the total number of patches on the whole tooth row. You can then take a value known as the OPCR average. And this is basically just taking all the teeth you have in your jaw, taking the total divided by the total number of teeth, 18. And that gives you the average complexity per tooth for that jaw. So in this case, we have approximately 8.4 average patches per tooth across our jaw. And I'm going to be presenting these average results for you all for the results. For our analyses, we use the Roboski et al. Fish Tree of Life for our tree. For our functional groups, we established these using diet data from fish basin and literature. We ran all of our analyses in our environment, so the package we use for OPCR is called Molar. And for our phylogenetic ANOVA, we use the RRPP method, using this because it's much less prone to type 1 error rates, and the method of permutation on residuals allows for much higher statistical power when comparing across different groups. So first thing I want to show you for results, first thing we actually did was looked at the number of teeth and how that relates to our tooth functional categories. And really what we saw was there was no influence of tooth function on the number of teeth along the jaw. Non-significant phylanova there. And one thing you might notice, so those sediment excavators and scrapers, they're sitting really low at the bottom there, hovering right around 1. Those are our parrotfish beaks, so we counted those as a singular tooth cutting surface. So that's why they're quite low there overall. But if you look at the other groups, there's really not a whole lot of patterning there across our different tooth functional categories. So really no influence of tooth function on the number of teeth is what we're seeing. However, when we look at our average results for OPCR, we see quite a bit of a different story here. So what we think is going on here is that fused beak dentitions are really driving up the tooth complexity. You can see there our sediment excavators, scrapers, And that really weird algal cropper way up there at the top, these are all our parrotfishes. And so they're really driving up that complexity overall. I'll point out the sediment scrapers, about 15 of those are parrotfishes. 4 of the algal croppers are also parrotfishes. And so you can see that those are really driving up the complexity, so much so you really can't see what's going on with these other groups as of yet. ANOVA is still non-significant, although trending a bit more towards significance, at least compared to the the tooth count data. And then another thing here, there's an independent transition to what's been called in the literature a beak-like dentition, although it is a non-parrotfish group here, and still these beaks are really driving up this complexity pattern overall. So now I'm going to show you the data with all the beak outliers removed to get a better look at what's going on there with the smushed data. And so when we pull away the beaks, we're really seeing pretty comparable levels of tooth complexity overall across our different tooth functional groups. Non-significant ANOVA. If anything, you're noticing that gray group, the shell crushers, they're showing some of the highest variation across our tooth functional groups overall. Algal croppers, the more kind of herbivorous clade, those are really no different from these other groups. So what we think is going on here is that transitions to fused dentitions are really what's driving up our tooth complexity. If you look at our tree here, the red circle shows where the parrotfish beak evolved, and you're seeing that massive pile of really complex dentitions all just in one transition there. You can see, highly complex dentition here with this fused parrotfish beak. At the top, that's that independent transition to the beak-like. There's 3 teeth there. Some of them are quite fused together. So those beaks are driving up the complexity. We see a few other complex groups here as well, but nothing, really compared to this massive spike in complexity for that singular fused beak dentition. So to wrap things up a bit here, how do we do with addressing our question? Well, we think that we have some mixed results here, right? So the beak dentitions are absolutely driving up the complexity. They are herbivores, or classically thought as herbivores. Although there are just minimal transitions here in our current data sets, and we did not find a significant difference in phylogenetic ANOVA. When we remove those beaks, there's really no difference being shown between herbivores and other tooth categories. And those durophages are showing some pretty high variation overall, some high levels, which is what's also been reflected in reptile studies too. For future work, we want to expand our sampling a bit more. So this is more kind of preliminary data. We want to expand to other independent transitions to different tooth functional groups, including beaks. So pufferfishes also have independently evolved a beak. They're doing quite different things in their beaks compared to parrotfishes. So we want to see if some of this drive and complexity is more associated with trophic novelties or the actual ecological functioning of what the fish is doing, as well as other more kind of piscivorous groups and more classically thought herbivorous clades as well. So with that, I'd just like to acknowledge the members of my lab for hearing me talk about fish teeth for the past 5 years. The Evans Lab for micro-CT scanning access, Dr. Keegan Melstrom for help with running OPCR analysis, excellent museum curators and collection managers for loaning me a bunch of specimens to do this work, and then funding sources, including the SSB grad student research award. Thank you. And I think I have time for a couple of questions, if anyone would like. Yes?
If the beak is essentially multiple fused teeth,
Mm-hmm.
can you adjust your count for the number of teeth that are fused to see how that compares to single tooth?
Yes, absolutely, and great point. So the parrotfishes, actually the beak is made up of a bunch of individual tiny teeth, so they are all fused. One thing with the method, I've ran into issues with running it to where if you assign, say if there's 30 teeth on that one beak, if you assign, 1,000 triangles per tooth, 30,000 triangles, it can sometimes overwhelm the molar package and actually will fail to run. There are some ways to account for that, but yeah, I've tried it both ways and just methodological consideration. Be happy to talk more about it after that. Thank you. Okay, I think that's my time. Next speaker is Maxwell Shafer.
Does this work?
Read buttons to the slides, red is to go back.
But do I have to click on mine?
Yeah, if you click on View Presentation.
Oh, perfect.
Yeah, and then download it.
Okay, hopefully this works. So thank you for coming. And thank you for the introduction. And I liked your talk as well. Very good. So I'm Max. So I'm coming from the University of Toronto, where I just started my research group. If you want to know more about me or my group, you can scan the thing or look me up. Or if you're watching this afterwards, you can do this, of course. And so today I want to talk about temporal activity patterns and sort of like long-scale evolutionary patterns. So the history of life on Earth has really been punctuated by these large-scale mass extinction events, right, that have occurred multiple times. And when these things happen, they really reshape life on Earth. And how this exactly works has been really a mystery. So during these events, you have large-scale elimination of lineages and species. The ones that do survive these events are then responsible or then seed the recovery of these events through like increased rates of diversification, right, and recovery. However, during these events, it's been well known that both the extinction and the recovery is very selective, right, and specifically so, where during these different events, specific lineages undergo extinction, and then the ones that survive, even specific ones of those, are responsible for the biodiversity recovery. And so this process is very mysterious. We have some insight into what's going on in certain mass extinction events, but not necessarily more general trends. And so, for example, one that's quite interesting is, the KPG mass extinction, which was responsible for the extinction of the dinosaurs. And famously, our mammalian lineages survived this extinction event and then were responsible for a lot of the biodiversity recovery. We became the dominant sort of terrestrial vertebrates. And it's been sort of understood and sort of recently nicely demonstrated that the mammalian species that survived this extinction event were nocturnal, and that following this extinction event, some lineages were able to transition into diurnal niches that were previously occupied by dinosaurs. And so from this, you can kind of get this sense that maybe the ability to transition into these diurnal niches was important for this recovery. But so what are these nocturnal and diurnal niches? So these are temporal activity patterns, right? So there are sort of four classic categories. Diurnal species, so these are the ones that are active during the daytime, they're sleeping at night. These include most bird species, most reptiles, a lot of insects, but also some mammals, including primates and us. You also have nocturnal species, which have the opposite pattern, right? So these are active during the nighttime and sleeping during the day. They have specialized adaptations for being active during the night. You have species that are crepuscular, so these are specialized for the dawn and dusk periods in between. And then you also have species that maybe you could argue specialized for being randomly active, so active during both the day and the night. So these are ephemeral species. And so why some species are nocturnal and diurnal is kind of a very complicated question, and we don't have really clear answers. But what we do know, in another recent work, Anderson and Wiens really looked at the evolution of these activity patterns across all tetrapods. So this is including amphibians, mammals, reptiles, and birds. And what they really found was a few things. One, that these activity patterns tend to be evolutionarily conserved. So lineages that are nocturnal stay nocturnal for long periods of time. And within these lineages, so within amphibians, within mammals, the majority of species there are nocturnal. They also found that transitions into diurnality within reptiles and within birds specifically was really associated with their evolutionary diversification. So being able to switch into those diurnal niches really allowed them to diversify extensively, and so this is kind of similar to what we see with the mammalian lineages that were able to transition. They also found that the last common ancestor of tetrapods, and the last common ancestor of a lot of these individual groups, was also nocturnal, and so this kind of suggests that nocturnality is this very long-lived behavioral pattern across the tree of life, but then being able to transition into diurnality seems to be important. So the dynamics here are a little bit still confusing. So tetrapods represent like a fraction of the vertebrate group, right? So if we want to consider these, I think the next step would be to really look at what's going on in the sister lineages to these groups, which are the fish, so bony and cartilaginous fish. So these represent, like we just heard, half of all vertebrate species. And so what we set out to do at the beginning of this project was to really ask, like, what is the evolutionary history of temporal activity patterns in fish, and how does that relate to tetrapods, and do we see similar things or different things in these organisms? And so unlike in mammals and in tetrapods, we didn't really have these existing databases of activity patterns, right? So I think this is a very underappreciated aspect, I think, of fish biology. And so we set out to generate this database ourselves through this meta-analysis of the literature. So we read through, between 5,000 and 10,000 papers and theses and all of these things. So I think if you ever have data, publish your thesis. It's really useful. There's lots of stuff in there, and people will eventually find use for it, I think, right? So it was mostly through Google Scholar, which allows you to access a lot of these things. And we have data from papers that either directly track the activity of these animals in the lab or in the field with like these satellite tags. You get these really nice data. But then we can also use proxies for activity like feeding rates or even in fish catchability. So the ability of the fish to be caught seems to be a proxy of their activity pattern as well. And so from this, we were able to collect data for about 4,000 species of fish. So it seems like a lot, but it's only a fraction of the total species. So it's about 10% of fish species, depending exactly on which list you use. And so what we could do, or what we thought we could do, is can we do an ancestral reconstruction of this? So can we look at this? And so that's what you see here. So this is these 4,000 species colored by their ancestral likelihood of diurnality in red or nocturnality in blue. We're pretty confident in our coverage in the sense that we cover, over 95% of fish orders, over 30% of fish families, and I said like about 11 or 12% of fish species. And from this analysis, we get sort of a few main conclusions. One, that the common ancestors of both cartilaginous fish and bony fish were nocturnal, so there's these blue points early in the tree. But that unlike the tetrapods, fish are really characterized by having both a lot of diurnal and nocturnal species. So they're about 50-50 in our data. I'm pretty confident that that's what really is happening. And we also have evidence from this reconstruction that there are hundreds of transitions between diurnality and nocturnality, so many, many, many more than in tetrapods. We can add this, so this is not a monophyletic group. So cartilaginous fish are the outgroup, and bony fish are actually the sister group to tetrapods, so we can combine this with the data that I presented from these other papers, and we can do this reconstruction across all vertebrates. This is a bit tenuous, it's very long time scales, but what this does support is this nocturnality in the common ancestor of all vertebrates, as well as in each individual group as well, of each major group of vertebrates. And then we can compare the number of transitions that occur, and so what you're looking at the top is just the raw number of transitions in our data, and then it's normalized to the number of lineages in our data, of course, because we don't have all of the species that are known. And you can see that for both bony and cartilaginous fish, they have many, many more transitions than the other groups, right? So they are transitioning much, much more. So they're much more able to transition. And so given all of these transitions, it then becomes interesting to ask, okay, so when are they happening, right? So we know in mammals that they happen mostly after KPG. So what this is now looking at this normalized rate again over time. And so if we look at mammals, we can see that the normalized rate over time, there's no transitions up until KPG, after which then they're able to transition. We actually see something very similar for amphibians, where we do see this increase in transitions after KPG. There is a blip before that I don't really understand what's going on, but at least around here there is an increase after KPG. If we look at the other groups, so sauropsids is reptiles and birds, right? So and bony fish, we see that there is also a similar kind of pattern where there's no transitions before a certain time point. And that time point is the JK boundary, at which point then they start transitioning, okay? And so this suggests that, for a long periods of time in their evolutionary history, they were not able to transition, and that something happened at these time points that then allowed them to transition. And so what we think is happening is that both of these time points are associated with elevated extinction rates. And so the hypothesis that we have right now is that the elimination of species through extinction is opening up these niches, which are allowing these lineages to transition, and that certain lineages are sort of more able to take advantage of this, right? So just to kind of sum up a little bit more, we have evidence for nocturnal species in the last common ancestors of all of these different groups, so mammals, tetrapods, and fish, and vertebrates in general, and that we think that certain lineages have an increased capacity to undergo these transitions, and particularly fish. So what you're looking at here is the percentage of lineages over time in fish that have undergone either 0 transitions, 1 transition, 2, 3, 4, 5 transitions. And you can see that the extant lineages, so on the very far right of the graph, about 90% of extant lineages have evidence of at least 1 transition, and we even have some lineages that have undergone 5 transitions, so nocturnal to diurnal to nocturnal to diurnal to nocturnal again over the last 400 million years. So we think that fish are really capable, really, really adept at doing this. Yeah, so just to sort of sum up a little bit more, yeah, so we think that this also kind of suggests, although it's a bit hard to really grasp and get around this, that potentially nocturnal species have some sort of advantage during extinction events, because we see this evidence that only the nocturnal lineages are surviving through at least these two periods in different cases in the different lineages. This could also mean that there's a disadvantage, particularly for diurnal species, but there's not really a sort of very strong or consistent hypothesis as to what might be going on there, although I have some theories. And that we do see this, that the nocturnal lineages are seeding the diversity recovery, right, through transitions, and particularly those lineages that are able to transition for whatever reason, either like molecularly, genetically, or ecologically, there is that opportunity and they're near close thing. And so this also suggests that it's not just mass extinctions that this could be playing a role in, but also anywhere where you're getting these new ecological niches, right? The species that can take advantage of both temporal versions or multiple temporal versions of those niches will potentially be more successful. And so another example of this, so this work is on bioRxiv, it's been on bioRxiv for a year, but we have other work also in cichlids. So another example of where this might be happening is in adaptive radiations, right? So where you have diversification of species because of adaptation to ecological niches. And so one of the most famous examples of this, of course, is the cichlids from the African Rift Lakes. Specifically, we worked on Lake Tanganyika, where you have hundreds of species that have evolved to and become adapted to these very specific ecological niches. And so we've done some work where we're really tracking these species in the lab, and we've identified really that there's also extensive temporal niche diversification happening in cichlids as well. And so we think that the cichlid, and in general, I think fish's ability to transition between these niches has really allowed them to take advantage of not only the new ecological niches, but also the different temporal versions of those ecological niches as well. And so this is maybe an underappreciated aspect of this, species diversification process. Okay, so I can finish by thanking. So I did this work while I was a postdoc in Alex Shear's lab, and we worked with the lab of Walter Salzberger. I worked really closely with another postdoc, Annika Nichols. She's on the job market now. She's interested in the evolution of nematodes and things like this. Lots of people in Walter's lab who are really great help, so some of you cichlid people might recognize some of these people. I've now just started my lab in Toronto. I have a very small lab, but if you're interested at all in working on these things, working with us, whatever, you can reach out or talk to me after. So I'm happy to take questions. I don't know if you want to use the microphone, but I saw this hand first.
Do you think, looking at your data, I wonder, do you think that there's been a lot of diurnal radiation, say, of mammals prior to the KC boundary, but then they all went extinct during the extinction?
Yeah, so I think that's a really good question. I don't know how to really prove it or look at this. Yeah, but I think definitely in the other lineages, I think for sure in fish this has happened because they've been around longer. So there was a longer period where they existed, and a lot of evidence from fossils that there was a lot of diversity there, probably also diurnal species. But it's really hard to have a clear proxy for this kind of behavior in the fossils. I think some people might want to use eye size, but I'm not really convinced that that is in general a good indicator of diurnality or nocturnality, right? So people say that bigger eyes, they're nocturnal, but there's also lots of evidence, especially for the lineages that have been nocturnal for a long time, like eels and stuff like that. They have very small eyes. They never evolved bigger eyes because they were never, diurnal, maybe. Yeah. Other questions in the back, I think, first.
Just one question. The first one, do you see a low, better survival for nocturnal because of the fact that the extinction event is related to a meteorite that just cleared the light, there is no more light in the atmosphere?
Yeah, I think that's a good explanation for what might have happened at KPG, but what might have happened at the other extinction events, I don't know. So you could also imagine temperature differences and things like this, but finding sort of a consistent thing that's happening at these different, and being able to really associate it with, it seems like this selection is happening at different times in the different lineages, so I don't really have an explanation for that either. But I think that's a good hypothesis, at least for that.
Is the fact to be nocturnal, is the pre-adaptation to be going to a lower depth?
Yeah, so in terms of fish.
Yeah.
I haven't looked at that. But I think that would be a good thing to look at. I think it's also in terms of like temperature and things like this, that's a big difference with depth. I don't know, I should look at it, yeah. It would be specifically in marine. I know there's not really an association with being marine or freshwater, so most of the depth differences would be in the marine environment, so maybe that's there, but I think I should look at that, yeah. I don't know if there's time for another question, but maybe we can talk after, yeah. But, yeah, maybe this one, yeah, because you had your hand up.
This is so cool and so exciting. I guess it's a super quick and not super well-formed question. A lot of the cichlid diversity is also associated with variation in color pattern and like this sexual selection effect. And I'm curious
Yeah.
about how like sort of this interaction with becoming nocturnal and maybe not having light to see those color patterns the same way that you think that interaction.
Yeah, so I think that's very interesting. I think one thing that maybe you might be interested in is some of the nocturnal cichlids have this very conspicuous and specific pattern, which is like this blue and black bars. And I've talked to some sensory ecologists, and they say that this is the most conspicuous pattern at night. So there's probably some association, but it might be more complicated, yeah. Are you next?
Yes.
Are you Callum? Yeah.
Yes.
I downloaded your presentation here. Maybe I can open it.
Well, I'm also going to talk about cichlids.
Yes, I'm very excited. So you just press the button.
All right. Wonderful. Thank you. Hello, I'm Callum. I'm a PhD student in the Department of Biology at the University of Oxford, supervised by Dr. Berta Verd and also Roger Benson at the American Museum of Natural History. So as a lab, we're really interested in understanding the kind of development of the teleostean vertebral column, for which we use kind of cichlids as a model for that. So thank you for introducing cichlids, my favorite fish by far. They're absolutely beautiful. Traditionally, the teleostean vertebral column is kind of divided into two domains, the precaudal, marked by the presence of these pleural ribs, so we can see with the pink here, which are essentially sort of very similar to thoracic vertebrae in mammals and other kind of tetrapods. These, of course, aren't tetrapods. And the caudal vertebrae, the tail vertebrae, ultimately, that lack these pleural ribs and they have hemal spines. But what's really interesting about vertebral counts and proportions is that we can use these as proxies for the developmental processes that give rise to the vertebral column. So there is a developmental process called somatogenesis, which all vertebrates go through, that form the segments that ultimately become the precursors of the vertebrae. So this is a zebrafish embryo. We can see that the tail extends posteriorly around the yolk sac. And we can see that these what are called somites are forming sequentially as the tail extends posteriorly. And so actually the vertebral count of the adults can alternately be used as a proxy for the number of somites that form during embryogenesis. And vertebral identity is ultimately provided by this super classical example of embryological gene expression by Hox genes, which pattern the anterior-posterior axis. So actually, by counting the number of precaudal and caudal vertebrae, we can have a good idea about how regionalization has evolved as these African cichlids have diversified, and also by taking a look at the count, we can have a look at how somatogenesis itself as an embryological mechanism has evolved over time. And so I study the whole African cichlid subfamily, and they have absolutely remarkable diversity in body shapes and, of course, in vertebral counts as well. But everything, the behaviors, coloration patterns, if there's a thing a fish can do, there's a cichlid that can do it. So some of my favorites are here. These Gobiocycla, for example, this remarkable rheophilic lineage buries itself essentially in the gravel of the bottom of the Congo Rapids. And they have a really cool vertebral column morphology, actually, which I'll talk about later. These Parasidini at the very top on your right, they're scale eaters. And endemic to Lake Tanganyika, they form schools of hundreds of fish, essentially just migrate around the lake looking for prey to bite the scales off. They have kind of lateral jaws as well to latch onto the side of their prey. I mean, truly remarkable. I mean, cichlids are amazing. And of course, as we've mentioned too, they are also known for their adaptive radiations. So the Great Lakes in the East African Rift Valley, so Lake Victoria, Lake Tanganyika, and Lake Malawi. So actually, because these are nestled within the wider subfamily, you get a really amazing model to also consider kind of how embryogenesis and kind of and how that relates to adaptive radiations as well. So really, my data collection has been very simple. This is outdated now because I have nearly 5,000 specimens of 570-something species. But the data collection is incredibly simple. I just count the number of precaudal, caudal vertebrae and also calculate a very simple body aspect ratio. So there is something, I mean, for me anyway, something very beautiful about this very, very simple data collection of just counting. And that is shown, that is very odd. I don't know what, there's clearly been something wrong with the upload. But I essentially put this into a big PCA plot. I apologize that the points aren't showing. But essentially, one thing that so the PC1, this variation of essentially increasing elongation, I apologize, it's negative, it's more elongate. I've now recently switched that, but never mind. And PC2 is actually regionalization of the vertebral column. So these two things are super important. The one thing we see actually is that the riverine species overall, which are in this kind of pink ring, actually have a very different axial morphology to their lake counterparts, so these ones that are in the lake radiations. So something about being in a lake environment is really important about driving elongation of the vertebral column. So overall, yes, the lake species are more elongate than their riverine counterparts. But actually what's really interesting is that they tend to have more elongate caudal domains too, relative to their precaudal domains. And what's even more interesting is actually this exploration of morphospace seems to correlate with the age of the adaptive radiation. So we have Lake Malawi in green, which is only really several thousand years old, and in blue you have the Lake Tanganyika adaptive radiation. I mean, there's multiple in Lake Tanganyika, but on the whole you're seeing that the more time you have, the more that you're exploring axial morphospace's ability to change your vertebral column. And one of the primary ways in which, again, why are my points not showing my plots? I'm very sorry about this. But that one is. One of the ways in which they are elongating themselves is by adding vertebrae, which of course makes sense. It's not the only way probably, because the actual amount of variation being explained, this needs to be updated, is about 47% rather. So actually it's not the only way, but it's definitely an important way. So elongate lineages are adding vertebrae in order to elongate themselves, to make themselves longer and thinner. But what's really interesting is that really, I mean, there are many different lineages that are elongating themselves, but one primary way that's convergently evolved is for kind of active piscivores to elongate themselves. So independently of what lake they're in, there are some riverine lineages that have done this. If you are piscivorous and you're an active open water hunter, you are elongating yourself. And which makes sense. You want to make yourself longer and slimmer if you're in open water because you move through the water column more easily, and so it's easier for you to catch fish. But actually there are other lineages that have elongated themselves. A lot of substrate dwellers and rheophilic lineages have also elongated themselves, probably as a means to bury themselves in substrate, for example. But what's very interesting is that actually there is, there's very little intraspecific variation in the vertebral counts. So this is very interesting in terms of development because it means that this process of somatogenesis isn't, there is some level of noise or plasticity in this developmental mechanism. But what's really interesting is that clearly the increasing your number of vertebrae is an adaptive trait, but the intraspecific variation hasn't varied between the lake rivers or radiations. So the root common ancestor of all cichlids had the same amount of variation, which is very strange, or sort of very strange. What's even more interesting is where you get changes in the total vertebral count, you don't change your intraspecific variation. So regardless of the number of vertebrae that you have the same level of Semitic kind of variability, which is very strange because we're not seeing a signature of positive nor negative selection. So really, it's a very odd problem. I mean, some of these lineages are diverging incredibly quickly. So our hypothesis was, well, maybe they've gained some sort of Semitic variability that they've been able to act upon. But it doesn't seem that they are doing that at all. So regardless of whether you have 25 vertebrae or 40 vertebrae, you're ultimately forming the same amount of plasticity as a species. You have the same amount of plasticity as a species. So it's incredibly strange. We don't know where this trait is coming from or whether we're even looking on the right scale in order to get this phenotype. And so we thought, well, really, actually, the thing that's being selected for here isn't the number of vertebrae, it's your body aspect ratio, because, of course, that is the thing that is being selected for in the environment. But it turns out if you look, so this species, Assotilapia calyptra, it's a riverine generalist native to Lake Malawi. It's also found in rivers connecting Lake Malawi. And it's also probably the sympatric ancestor of all Lake Malawi cichlids, although that's hotly contested, of course. But we thought, oh, maybe, just adding one extra vertebrae is enough to give you some sort of significant increase in body aspect ratio. It doesn't. Again, the points aren't showing, which I apologize for. And this 28 is probably not an Assotilapia calyptra. So it's very, very strange. Maybe, and again, some of these lineages are elongating themselves within thousands of years. I mean, this is not something that's taking millions of years. So really, there's only 3 ways you can change the structure of your vertebral column from an ancestral state following diversification. And we're ignoring changing the vertebral aspect ratio for now, so the actual dimensions of the vertebrae themselves. So from an ancestral state, you can have what we've termed pure homeotic shifts, where essentially you maintain the number of vertebrae, but you change the regionalization. So you change the number of precaudal or caudal vertebrae. There are pure Semitic changes, where actually you just change the number of vertebrae, but you maintain the ratio of precaudal to caudal vertebrae, or you have what we've termed combined effects, where you're changing both. And so actually, if you detect a lot of signature for combined effects, what we're actually saying is that semitogenesis and regionalization, these 2 developmental mechanisms, are evolutionarily uncoupled and modular. And so, again, the points aren't showing. What we should be seeing is that actually, we're looking at the phylogenetic independent contrasts for the caudal count and the precaudal count. We should be seeing that actually all of the points are distributed across this space. So actually what we're trying to say is that combined effects have dominated during the diversification of African cichlids. So actually, on the whole, there's been kind of major evolutionary uncoupling of these 2 mechanisms, which actually doesn't make it. It raises some very, very interesting developmental biology questions about how those 2 processes interact, because there's 2 schools of thought. One is that semitogenesis and Hox patterning have to be intimately linked and molecularly linked, and the other is that they're not linked at all. It's just a matter of time, and they just balance each other out, more or less. So actually what we're saying is, well, no, evolutionarily it seems that it's the second hypothesis, is that they don't have to be intimately molecularly linked. Which I don't know if there are any developmental biologists in the audience, but it's one thing that we're struggling to abstract, actually, and understand more. So, to summarize, riverine and lake cichlids are being driven to adopt different axial morphologies, and I'm happy to talk more about that, because there are lots of different reasons why that might be. And in fact, the common ancestor of all African cichlids, I've reconstructed the ancestral state. It seems to be this very deep-bodied, very short, very few vertebrae species, which might be actually really important for people looking for cichlid fossils. So if you're a cichlid fossil hunter, please come and talk to me. Despite vertebral counts being highly evolvable, intraspecific variation is low, and elongate lineages have not lost or gained kind of what we've termed Semitic variability, or Semitic sort of plasticity, but I don't like to use that word. Changes in total count are usually accompanied by a shift in regionalization, suggesting semitogenesis and Hox patterning are highly evolvable and evolutionarily uncoupled. So future work which is now ready, is going to be published. I found out yesterday, which is very exciting. I've generated a whole-body micro-CT scan dataset of Lake Malawi cichlids of 56 species, 116 individuals from 26 genera, so massive coverage of the radiation, which includes the heads as well for people that are interested in the anterior of the fish. But this was to look at vertebral shape as an adaptive trait. I have some hypotheses about how vertebral shape might have been modified during the Lake Malawi radiation. and maybe trying to use that as a means to look at the evolutionary dynamics of morphological variation in the radiation, and whether we can sort of look at generalizable rules for adaptive radiations. I've also, a lot of my PhD actually has been sunk, not even sunk because I absolutely love it, into generating interspecific hybrids to do some QTL analysis. So these are representatives of my F1 clutches, which are now breeding and making F2s. So we can do QTL analysis. They're incredibly beautiful, by the way. We can do some QTL analysis. There's loads and loads of different traits that we can investigate. Dentition is one, for example. Jaw morphology, craniofacial morphology, lots and lots of different things. So if you're interested in collaborating, we desperately need collaborators to do this F2 QTL analysis. And my PhD finishes in a matter of months. So please come and talk to me about that. But just to acknowledge my funding, I'm funded by the BBSRC in the UK, and I got funding from my college as well. And it takes a village to raise a PhD student. So a lot of people have been involved in this. So thank you very much. And this is our update. Yes.
That was a great talk. So I wonder if you're seeing any sex-specific differences for the elongation, particularly with that rate, because a lot of this is looking out through.
Yes.
So yeah, I'm wondering if maybe in the data set that you created, if that's a variable that you're going to look into.
So I don't know about sex differences because a lot of it's been on museum specimens, and so it's very difficult to sex cichlids. I mean, there are ways but without their coloration patterns, it can be incredibly difficult. However, what I do know is that brooding behavior doesn't seem to affect the structure of your vertebral column shape. So actually, mouth brooders, substrate brooders, I've compared them all, and it doesn't seem to be a major influencer.
That's quite interesting.
Yes.
So it's more about their diet and their
Environment.
and stuff
Yes.
rather than the current.
Yeah, precisely. No worries, thank you. Yes.
Yeah, that was a great talk. I was
Thank you.
wondering about the shape differences that you briefly mentioned. Because like in mammals, you have some of the vertebrae which have been enormously elongated, like giraffes, for example.
Yes.
Is this happening, or is this not happening, and you think there's a constraint because of their mode of locomotion, like using the primary body axis for locomotion?
Yes, so it seems that in teleosts in general, modifying your vertebral aspect ratio doesn't seem to happen. So there's a few examples. The barracuda, for example, very elongate fish, but actually has very few vertebrae because all they've done is elongated their vertebrae. But that probably restricts your movement, so it makes your vertebral column very stiff. Whereas I think if you're adding, so with eels, for example, they've added vertebrae because they want to be flexible, because I think one of the hypotheses is that because they're burrowing, they need this kind of flexibility. I'm in the process of checking whether the vertebral aspect ratio is different between lineages, but I doubt it is. What I do think is different is the overall 3D shape of the vertebrae. So I think pelagic lineages, for example, have much squarer, flatter kind of vertebrae, for example. Thank you.